Hi everyone, I'm Manuela Costa, postdoc at the Laboratory for Clinical Neuroscience in Madrid. I'm very glad to be here today at this conference for presenting the last results about memory formation in humans. Sorry. Can you see the screen?
No.
Sorry, it's just the slide didn't change. Is it fine now? All right. Okay, so as we all have experienced, memory of emotionally arousing events tend to be more vivid and to persist longer than neutral ones. But why this happens? Well, we know the amygdala is an important area for emotion regulation, so it's not surprising that the amygdala also play a fundamental role in emotional memory formation. Accordingly to the memory modulation hypothesis, indeed, arousing experience activate noradrenaline release that will modulate memory in other brain regions, such as the hippocampus or the cortex. But which is the mechanism of amygdala and hippocampus communication during emotional memory formation? This is the main topic of our study. Previous findings have suggested that amygdala-hippocampus communication might occur via coordinated oscillatory activity. But today, I will try to show you that other mechanisms, such as the exact phase at which the coupling occurs, can be important for emotional memory formation. For this study, we recorded electrophysiological activity from 15 patients recorded at the Ruber Hospital in Madrid and at the University Hospital in Zurich. All patients had electrode implanted in the amygdala, and 8 of them also had electrode implanted in the ipsilateral hippocampus. Patients perform a memory task. The first day, they saw either unpleasant or neutral scenes, and they were asked to make an indoor and outdoor judgment. After 24 hours, we make the retrieval, and patients were asked to say whether they remember, they were familiar, or they don't remember the picture. We start analyzing the behavioral response, and in line with previous findings, we actually found that recollection performance was higher for emotional stimuli compared to all other types. In other words, patients remember better the emotional items compared to the neutrals. So now we can look at the oscillatory activity in both structures. I want to stress that we focus on the subsequent memory effect, meaning that we divide a trial at the encoding whether they were later remembered or forgotten. And we also collapse familiar and forgotten activity. So looking at the amygdala activity, we actually found a fast gamma oscillation that was associated with successful encoding of unpleasant scenes. This result was consistent when we look at the 8 patients that also had the electrode in the ipsilateral hippocampus. By contrast, when we look at the hippocampal activity, we actually found that the gamma response was associated with subsequent recollection independently on whether they were emotional or neutral. So just to summarize, in the amygdala, we found faster response starting at around 400 milliseconds, while in the hippocampus, we found a slower and later response that starts around 500 milliseconds and that was not sensitive to the emotional content of the scenes. Now, having characterized the activity in the amygdala and the hippocampus, we moved to study the coupling between these two structures. From previous study, we know that there is, during retrieval, they found amygdala-hippocampus theta synchrony in rodents. And in non-human primates, during an adversarial learning task, they found a flow of information going from the amygdala to the cortex, still in the theta range. In our dataset, we didn't find a significant difference in the low frequency across conditions, but as you can see, theta was present in all trial types. So we calculate coherence and Granger causality analysis. I mentioned already that we didn't find any type of coherence, but when we look at the Granger causality, we actually found that the amygdala theta leads the hippocampal theta when patient viewed emotional scenes, with a lag of around 24 milliseconds. Phase amplitude coupling has been proposed as a mechanism for facilitating memory formation. So we reasoned that we could find a difference between successful and unsuccessful encoding of unpleasant scenes. We perform the modulation index between the phase of the amygdala theta and the amplitude of the hippocampal gamma. But actually, what we found is that phase amplitude coupling was evoked to all emotional stimuli, but was unrelated to memory. So we reasoned that modulation index is actually not sensitive to determine differences in the preferred phase at which the gamma coupling occurs. Look, for instance, this example. These are two examples of phase amplitude coupling, but in one case, the peaks occur in the peak of the theta oscillation, and in the other one on the throw. So we develop a new matrix that we can call phase amplitude coupling opposition index, which quantify the strength of phase opposition between two conditions. With this approach, we actually found that the magnitude of gamma amplitude was concentrated at different phase bins between successful and unsuccessful encoding of unpleasant scenes. Here on the x-axis, you can see the bins of theta, and this is the amplitude. So you can see that successful encoding of unpleasant scene that is in red is more localized in this preferred phase compared to emotional scenes that were not later recollected. So in other words, this result means that the precise amygdala theta phase at which the hippocampal gamma peaks occur is important for successful encoding of unpleasant scenes. Just to summarize, if you remember, we found that amygdala gamma response were selective for successful encoding of unpleasant scenes. And now we are showing a phase-dependent coupling between amygdala theta and hippocampal gamma. I want to stress that if there is phase amplitude coupling opposition, also there should be a delay between the two gamma oscillations. So we explored these relations, and in particular, we calculate a correlation between the peaks envelope of gamma oscillation in the amygdala and the hippocampus. As you can see from the plot, we actually found a significant transient coherence when the amygdala peak precedes the hippocampal ones. And finally, we found that there was a proportional lag during successful encoding of unpleasant scene between transient coherence, that you can see here, and the preferred phase delay, that means the result from the phase amplitude coupling opposition converted in the delay, showing, as I said, that there was a proportional lag between the two structures. So I hope that I convince you that emotional memory formation is actually phase dependent, and particularly that the specific phase at which the coupling occurs is fundamental for this process. Moreover, with our data, we show how the amygdala is influencing hippocampal oscillation during emotional memory formation. Before ending, I want to thank my colleagues, my supervisor, Brian Strange, Diego Lozano Soldevilla, that is also first author in the paper, all my colleagues, the hospital, and the patients that took part in our experiment. And thank you for watching.
Okay, thank you very much, Manuela. So we don't see many questions, but while people are making up their minds, well, it's been a wonderful talk, and I have a curiosity really, and trying to know a bit more of these mechanisms that may be altered in some pathologies that have over-representations of, for example, bad memories, let's say, anxious people, or I don't know, different types of diseases that may have too much of an enhancement of bad memory. How would this mechanism work? Would the hippocampus compensate, or what would be the way out?
Yes, thanks, Ignacio, for your question. Actually, yes, we think that our result can be relevant for some pathologies, such as the post-stress traumatic disorder. I mean, remembering negative experience is important for survival, but as you said, it can become problematic in this type of pathology. So we think that our mechanism can be interesting, especially related with the possible stimulation that can be applied to this type of patient. There are already some cases. So I think, sure, it's relevant for this type of, for the clinical aspects.
Okay. We have, okay, so that's quite interesting. In fact, I see there are no more questions going on, so